Alright, so this is the second video following the one where I explained how we do initial catalase tests to differentiate between staph and strep, and then within the staph we do a coagulase test and a mannitol test to differentiate between Staph aureus versus Staph epidermidis. But now we're talking about the strep bacteria, and there are many different strains, and what are the tests we do to identify which one is which. So the first thing we need to establish, and this is important, the first thing we need to establish is type of hemolysis. So hemolysis is the property of being able to burst red blood cells, right? So when bacteria are invading a host, some bacteria have this property that actually make the cells burst, the red blood cells. Hemo is referring to blood, and lysis is the disintegration of the cell. So this property is a factor that makes the bacterium even more virulent. So when we plate these cells on a plate where the agar contains red blood cells, so this is all red blood cells inside, right, inside the medium, red blood cells, and we plate our cells on top, on the surface, what we might see is around the cells we will see a halo a halo where hemolysis took place, a white halo where hemolysis took place. So around the cells, this is where the red blood cells burst, and we do not have that red pigment anymore, and that visually it allows us to see where hemolysis took place. So we have different types of hemolytic strength, and the first one I'm going to talk about is beta hemolysis. So we have to identify first if the strep is beta hemolytic, alpha hemolytic, or gamma hemolytic. So beta hemolysis refers to the strongest hemolysis. So hemolysis. So this is where we will have that white halo. It will be very stark contrast between the cells growing on the surface and missing pigments all around these cells, because they destroyed all of the red blood cells inside the medium around them. So the strongest hemolysis is beta. For me, beta sounds like burst, and this is the strongest hemolytic property. Alpha is less strong. So when you look at cells on the surface of a plate, and they did have some hemolytic effect on the red blood cells in the plate, there is a halo around them, except it's not a complete breakdown of the cells, and we have a greenish halo. It's odd. It's because it's not completely hemolyzed. We have remnants, and we have this kind of a greenish halo to that circle of damaged cells around the streps, right? We're talking about the streps on the surface of this blood plate. So that takes some practice to kind of see the difference between the two. And then finally, gamma is no hemolysis. So we would see the strep cells completely surrounded with red red blood cells on the surface of that red plate. So first we have to establish if our mystery strep is beta, alpha, or gamma. So you look at the plates, the red plates, red blood plates, where the cells are growing, and you establish that. Once you have established that you are dealing with one or the other hemolytic property, you have to concentrate on each one because the test will be different. So if you have a beta hemolytic strep, for beta hemolysis, what you need to do is you need to look at resistance or sensitivity to bacitracin. So this is an additional test. We put antibiotic filter, so bacitracin. I don't arbitrarily. I'm using this acronym BT. This is the filter. So this is the filter with the antibiotic in it, and there's a whole plate of cells, right? These are strep cells. We still don't know what kind, what strain, but this is bacitracin filter. So now, if it's sensitive to bacitracin, we are going to have that as being group A strep. And what that means is that you have the filter, then you have your cells, but these cells, these group A cells, were actually sensitive to that antibiotic. And there is a halo, so now we're talking about a different kind of halo. This is a halo of dead cells, right? So sensitive means we have dead cells here around the antibiotic. So this is live cells and dead cells. Okay, if they are not sensitive, if they are resistant to the antibiotic, we do an additional test that we call the camp factor, which I will explain in a different video. The camp factor tests. And that usually comes out positive. And the camp factor is a factor that some cells have that makes the hemolysis even stronger. So it's and these cells that have this camp factor are called group B strep. All right. This is for beta hemolytic streps. So let's move on to alpha. Alpha. If we are dealing with alpha hemolytic strep, that means the hemolysis is not as strong as beta, but we still have some hemolysis. And what we test is we test resistance or sensitivity to uptoken. So this is another antibiotic, and if we find the cells to be sensitive to uptoken, we have identified that this is strep pneumoniae. Strep pneumoniae. So again, here's there's uptoken in a filter, and then uptoken has killed, because sensitive means they are killed cells by uptoken, and then the rest is live cells. But if they're sensitive to uptoken, that means we're dealing with strep pneumoniae. If they are resistant to uptoken, then we're dealing with alpha strep viridians. So alpha strep viridians will continue to grow even in the presence of uptoken because they are resistant to that antibiotic. Okay. Last scenario is if you have a gamma hemolytic property. If you have a strep with a gamma hemolytic property, then you do a bile esculin test. Bile esculin test. So esculin, so we put, if esculin is produced by the cells, esculin in the presence of ferric citrate, which is available in the medium as iron. Oops, I'm writing faster than my pen. Ferric citrate. So in the presence of ferric citrate, which is iron in the medium, we're going to end up having a black precipitate. Okay, so some cells produce esculin, and the presence of esculin combined with the iron in the medium will create this black precipitate. So if we have a black precipitate, that means we have a positive test. The black precipitate. So when we have a positive test, there's one more step before we actually determine which bile esculin positive strep strain it is. We have to grow these still mystery strains in salt broth. So salt broth, growing in salt is actually very difficult for bacteria, and many of them will not grow, but some of them will. And if there is growth in salt broth, then we are dealing with group D Enterococcus. And if there is no growth in salt broth, then we are dealing with group D non-Enterococcus. So both group D are bile esculin positive. Both group D are bile esculin positive. That means these strep cells produce esculin, and the way we can visually see that is if we add iron into the medium, that esculin combines with it and creates this black precipitate. So you will see a black precipitate following a test with bile esculin. But to identify if it's Enterococcus or non-Enterococcus, we do an additional test of growth in salt broth. So we put the mystery strep in salt broth and we see if there is growth after 48 hours. Is the salt broth still clear, or is there cells floating there? We also do a CAMP test. We check the CAMP test. We check the CAMP test on cells like that because sometimes we have, so if the CAMP test comes out positive, we're actually dealing with a group B strep, which is something we saw with the alpha hemolysis. Well, we actually have group B gamma hemolytic cells with hemolytic property, and we can only really see that if we also do the CAMP test. These group B strep cells have the CAMP factor, and that's really what allows us to pull out these group B cells. But in this case, they're group B cells, but they have a gamma hemolytic property as opposed to the alpha hemolytic property. So that's it. So determine the type of hemolysis first, and then depending on the type of hemolysis, you have either a test with an antibiotic called bacitracin, or you have, if you're alpha hemolytic, we will test you on uptoken, and if the strep is gamma hemolytic, we'll do bile esculin, growth in bile esculin, and see what the results of that will be. And that's it.